Welcome student for today's lecture. The students, in last lecture we have seen about the Griffith's experiment. Last step, Griffith observed that there is something which is transferred from the S type strain to the R type and that is due to which here from the heat killed S type strain into the R type strain something is transferred and due to which here the death of the mice takes place. Okay, so first we see that is about the Griffith experiment in the
experiment. Griffith in the year 1928 was working on a bacterium known as Streptococcus pneumonia. Streptococcus pneumonia has two types of strain, smooth strain and the rough strain. Cells of the smooth strain have a protective layer known as the capsule, which protects the cells against the immune system. This makes the smooth strain highly pathogenic. The rough strain lacks capsule. Hence, the immune system can easily kill the rough strain. When the cells of the smooth strain are injected in mice, the mice is killed. Whereas when the cells of the rough strain are injected in mice, the mice survives. In the next experiment, the cells of the smooth strain were killed by heat. When these cells are injected in mice, the mice survives. However, when the cells of the heat-killed smooth strain are mixed with the rough strain and injected in mice, the mice is killed. And this result was surprising. This means something was getting transferred from heat-killed S strain to live R strain, which converted the live R strain to S strain, which was highly pathogenic. And this phenomenon is known as the transforming principle. Griffith did not know what is getting transferred from the heat-killed S strain to the live R strain. Further experiments to find the transforming principle was done by Avery MacLeod and McCarty, and they showed that the transforming principle was the DNA.
So in the Griffith experiment, that is, we have seen the fourth step of the Griffith experiment. So see, that is which is the fourth step of the Griffith experiment is. So in the fourth step, that is, there is the mixture of heat killed S type of the strain, heat killed S type, fourth step of the experiment, heat killed S type plus R strain. And this mixture, when injected to the mice, the mice gets dies. Mice gets die. So here, in this fourth step of the Griffith's experiment, that is here, from the heat killed S type bacteria, there is one substance that is transferred into the S strain. Okay, and due to which here the mice gets dies. So here the Griffith who isolate the blood sample of this mice. Isolate blood sample of the mice and observe that is in this blood sample of this mice there are the S strain, that is the S type of the bacteria. S type of the bacteria. So here this S type, that is the pathogenic. It is the pathogenic, and here this S type bacteria, these are the pathogenic, and due to which here the mice gets dies. This mice gets died. That is the fourth experiment. So here in this fourth step, that is from the heat killed S strain, the substance some transfer into the R strain, due to which here the R strain bacteria, it get transformed into the S type. And here in the blood sample of these mice, there are the S type bacteria. So here the R strain, it get transformed. R strain get transformed into the S type. R strain get transformed into the S type. So here the Griffith, who conclude that is there is something which is transformed from the S type, heat killed S type, to the R strain, and here the something which is transferred from the heat killed S type to the R strain, and due to which here the R strain bacteria get transformed into the S type. So transformation means what? That is, here the one bacterial strain, it get transformed into the another bacterial strain due to some change, that is here due to some genetic change in genetic material. आणि मग इथं ट्रान्सफॉर्मेशन हे आर स्ट्रेनचं एस टाइप मध्ये कशामुळे झालं? सो देर इज समथिंग, बट ग्रिफिथ who does not prove which is the transforming principle. So here who proved that is there is one transforming principle. There is one transforming principle. So this transforming principle, due to which here R strain bacteria transform into the S strain. R strain is the non-pathogenic. So this non-pathogenic bacteria, it transform into the S type, that is the pathogenic. आणि हे S type मध्ये ट्रान्सफॉर्मेशन झाल्यामुळेच हे बर्ड माइस काय होतो? डाय होतो. That is mice causes the pneumonia. So here the Griffith, who conclude that there is one transforming principle, but the transforming principle, which is the transforming principle? Is it is DNA, is it is RNA, or it is the protein, or it is the carbohydrate? It was not proved by the Griffith, which is the transforming principle, who only told that there is the transforming principle, and due to which here the R strain, it get transformed into the S type. R strain get transformed into the S type. So here the three scientists after the Griffith experiment is that is who proved that is transforming, there is transforming principle, that is in 1928. But after the Griffith, after the 10 years of the Griffith, that is in 1944, the three scientists who proved that is which is the transforming principle is. So here the scientist Avery MacLeod and McCarty, these are three scientists who proved that is which is the transforming principle. So these are three scientists who proved that is by the using the Griffith's experiment, that is here one, that is the heat killed S type bacterial strain. These three scientists use the three substance from the heat killed S type bacterial strain, and they add that is R strain and observe that is which is the transforming principle is, which is the transforming principle is. So here the Avery MacLeod and the three McCarty, these three scientists, these are the microbiologists. So these three scientists, by the experiment, that is by the test tube assay experiment, they proved that is the which is the transforming principle is. So for their experiment, they use the heat killed S type bacteria, and from this heat killed S type bacteria they isolate the protein, RNA, DNA, and then observe that is which is the transforming principle is. So see, that is which is the about the Avery MacLeod and McCarty experiment.
Streptococcus pneumonia has two types of strain, smooth strain and the rough strain. The smooth strain have a protective capsule, as a result of which they are highly virulent, whereas the rough strain is not virulent. In the year 1928, Griffith discovered that when heat-killed smooth strain is mixed with the rough strain and injected in mice, the mice died. This result was surprising. It means something was getting transferred from heat-killed S strain to live R strain that made the rough strain virulent. This phenomenon is known as the transforming principle. Griffith did not know what was getting transferred from heat-killed S to live R strain. So in the year 1944, Avery MacLeod and McCarty did a series of experiments to find the transforming principle. They took cell lysate of heat-killed S strain and treated with different enzymes. Once the respective enzyme treatment is given, the mixture is treated with R strain and incubated, and finally spread it on Petri plates to see what happened to the rough strain. The presence of smooth colonies means that the rough strain got converted to smooth strain. It was only DNA's treatment that failed to convert the rough strain to the smooth strain. This means that the DNA was the transforming principle.
The Avery MacLeod and McCarty experiment. So in 1944, these three scientists, Avery, MacLeod and McCarty, these are three scientists who proved that is the DNA is the genetic material. So here these are three scientists who isolate the blood sample of the heat killed S type of the bacteria. म्हणजे ग्रिफिथ एक्सपेरिमेंट मध्ये जे हीट किल्ड एस टाइप बॅक्टेरिया आहेत त्यातल्या तीन जे काही पार्ट्स आहेत म्हणजे जनरली आपण त्यांना काय म्हणू थ्री थिंग्स which are isolated by the Griffith. That is here the three things. These are isolated from heat killed S type bacteria. Heat killed S type bacteria. So here these three things, first is the protein. Second is the RNA and third is the DNA. First, from the heat killed S type bacteria, isolate the protein, RNA and DNA. Then here, first in protein, here in protein he added the R strain, R type bacteria, that is which are the rough bacteria, and observe that is here which type of the bacteria are seen. So there is no change in the bacteria, that is here the R strain which remains as it is. R strain, it never get changed, that is R strain which remain as it is. R strain remain as it is. Then in the RNA, that is it is isolated from the heat killed S type bacteria. So here RNA, in RNA here the S strain of bacteria are mixed, and observe that is in this mixture also here the R strain bacteria which remains as it is. That is, there is no effect of the on the this R strain bacteria, that is they are rough, so which remain R strain. This R strain, that is rough bacteria, remain as it is. That is, here the R strain bacteria, it does not show any change. It does not show any change. So here in the mixture of protein and the R strain, that is here the R strain which remains as it is, then added the RNA and here R strain. In this mixture also the R strain bacteria which remains as it is. Then in the DNA, he added R strain bacteria, that is which are the rough bacteria, and observe. So in this mixture he observed that is R strain, it get transformed and convert into, that is the S strain. So here this R strain bacteria, it gets capsulated, that is it produces the wall around itself and it becomes the capsulated. It becomes the capsulated, which is the virulent, that is the pathogenic. Which is the virulent, that is the pathogenic. And here these three scientists, that is Avery MacLeod and McCarty, these three scientists, by this test, who proved that is here the DNA is the transforming principle. Okay, due to which here the R strain, it get transformed into the S strain. That is the R strain bacteria which produce the wall around itself, it becomes capsulated, it becomes pathogenic. So here the Griffith, who by his experiment, who proved that there is one transforming principle due to which the R strain bacteria, that is in the mixture of heat killed S type and the R strain bacteria, here the R strain bacteria, that is it becomes capsulated and here it converts into the S strain. It converts into the S strain. So see, that is student, here the scientist Avery MacLeod and McCarty, who proved that is the transforming principle is the DNA. Transforming principle is the DNA. म्हणजे कोणते ट्रान्सफॉर्मिंग प्रिन्सिपल आहे इथं? DNA is the transforming principle. Okay. So here what is meant by transformation? That is, transformation means here the transfer of the something that is from that is the genetic material from the one bacteria to the another bacteria, that is known as the transformation. So here the transformation takes place, that is here the substance, that is the DNA. So DNA is the transforming principle. So conclusion of the Avery MacLeod and McCarty, that is these three scientists who proved that is the transforming principle is DNA. So here they proved that. So after these three tests, that is they use some digesting material, that is the three digesting materials which can be digest the protein, RNA and the DNA. So here these the three scientists who use the enzymes. So there are the three enzymes are used by these three scientists. See, that is to digest the protein, they use the enzyme, that is the proteinase enzyme. Proteinase enzyme, these are the digest the proteins. these digest the proteins here. The one enzyme, that is the proteinase. One enzyme, that is the proteinase. This proteinase enzyme which mainly digests the protein. Second, the RNase enzyme. This RNase enzyme, it mainly digests the RNA. And third, that is the DNase enzyme. It mainly digests the DNA. So here these are three scientists, Avery, MacLeod and McCarty. These are three scientists who use the three enzymes. And by using this enzyme here, the proteinase enzyme when used, that is the protein can be digested. If they use the RNase enzyme, then RNA can be digested. And if use the DNase enzyme, then DNA can be digested. So in last step, we have seen, that is in last step of the Avery MacLeod and McCarty experiment, here the DNA plus R strain. Okay, DNA plus R strain, and here the R strain, that is it get transformed into the S strain. So here we have seen, that is the R strain, it get transformed into the S strain, that is the DNA is a transforming principle. Okay, so they use the DNase enzyme. म्हणजे नक्की DNase transforming principle आहे की काय, यासाठी त्यांनी काय केलं तर यातला DNase आपण डायजेस्ट करू, DNA डायजेस्ट करू आणि त्यानंतर ऑब्झर्व करू की नक्की R strain चं कन्वर्जन हे ट्रान्सफॉर्मेशन S strain मध्ये होतंय की नाही. ठीक आहे, मग त्यांनी काय केलं तर DNA प्लस R strain प्लस एन्झाईम यूज केलं DNase. एन्झाईम यूज केलं DNase आणि हे सगळं त्यांनी काय केलं तर एका टेस्ट ट्यूब मध्ये घेतला. टेस्ट ट्यूब, सो सी दॅट इज इन दिस टेस्ट ट्यूब. Here the DNA is there, then the R strain, that is the R strain, rough strain bacteria, it is the R strain, and they use the DNase in this, that is the DNase enzyme. So all this mixture is used in this test tube. Okay. And observe that is here the DNase enzyme digests the DNA. So here in this test tube here the R strain which remain as it is. That is there is never change in the R strain. So here the R strain which remain as it is in this test tube assay. म्हणजे जर इथं हा DNA जर काढून टाकला म्हणजे डायजेस्ट केला या DNase एन्झाईमनी, तर हा R strain जो बॅक्टेरिया आहे, रफ बॅक्टेरिया, which remain as it is. That is there is never change in the rough strain. So here in this mixture, that is a DNA and R strain. So here DNA is the transforming principle, and due to presence of this DNA, here the R strain which get transformed into the S strain. Okay. So here the transforming principle, म्हणजे ग्रिफिथने काय सांगितलं, there is one transforming principle, that is which mainly transfer from the heat killed S type bacteria to the R type, and due to which here R strain it get transformed into the S strain. बरोबर आहे. मग त्यापैकी अवेरी मॅक्लेड आणि McCarty यांनी त्या तीन सायंटिस्टने काय केलं बघा, या तीन सायंटिस्टने काय केलं तर हीट किल्ड S टाइप बॅक्टेरिया जो आहे त्यातला एक एक थिंग बाजूला घेतली. म्हणजे सुरुवातीला प्रोटीन घेतलं कार्बोहायड्रेट घेतलं आणि RNA घेतलं, DNA घेतलं आणि मग प्रोटीन आणि R स्ट्रेन हे एकत्र करून बघितलं की त्या R स्ट्रेन मध्ये काही बदल होतो का? R स्ट्रेन मध्ये बदल झालेला नाही. त्यानंतर त्यांनी RNA घेतलं, हिट किल्ड यस बॅक्टेरिया मधला आणि काय केलं, त्यामध्ये पुन्हा R स्ट्रेन टाकला आणि ऑब्झर्व केलं, R स्ट्रेन आहे असेच दिसले. मग त्यांनी काय केलं, DNA घेतलं आणि R स्ट्रेन यांचं मिक्सर घेतलं आणि मग मात्र त्यांच्या लक्षात आलं की R स्ट्रेन बॅक्टेरियाचं ट्रान्सफॉर्मेशन कशामध्ये झालं, S स्ट्रेन मध्ये. आणि म्हणून काय सांगण्यात आलं, DNA इज द ट्रान्सफॉर्मिंग प्रिन्सिपल. पण नक्की DNA हे ट्रान्सफॉर्मिंग प्रिन्सिपल आहे की नाही हे ऑब्झर्व करण्यासाठी त्यांनी काय केलं, तर DNase एन्झाईम यूज केलं की जे डायजेस्ट काय करू शकतं, DNA डायजेस्ट करू शकतं, बरोबर आहे. म्हणजे इथं DNA ची ऍक्टिव्हिटी जी आहे ती काय होईल, स्टॉप होईल ड्यू टू द एन्झाईम DNase. आणि या DNA ची ऍक्टिव्हिटी स्टॉप झाल्यामुळं इथं R स्ट्रेन जो आहे हा R स्ट्रेन याचं मिक्सचर जेव्हा यूज केलं, DNA R स्ट्रेन आणि DNase एन्झाईम एकत्रित, तर R स्ट्रेन हा आहे असाच R स्ट्रेन राहिला. म्हणजेच DNA इज द ट्रान्सफॉर्मिंग प्रिन्सिपल. So here the conclusion of the Avery MacLeod and McCarty experiment is what, that is they conclude that is the DNA is the transforming principle, not the protein or not the RNA. So okay student, so today we stop here.